Welcome to Innovation Day 2023, presented by Connect. We've got such an incredible array of things happening here, supporting the life sci and tech communities in San Diego.
So guys, we're going to finish with a bang, and I'm so honored to introduce to you Dr. Craig Venter, who's, of course, renowned for his contributions to genomics, including sequencing the first human genome and constructing the first synthetic bacterial cell, among many others. Craig is a recipient of the 2008 National Medal of Science and a member of the National Academies of Sciences. What's fun is Craig is going to have a fireside chat with Ashley Van Zeelen, who, among her many accolades, was the CEO and co-founder of Cipher Genomics, which was acquired by Human Longevity, co-founded by Craig. So I love the circular logic here. And they're going to be talking about the exponential advances in genomics and how we're moving from reading to writing the genome. So without further ado, great to see you.
Can you hear me? Okay. I can. Well, thank you all for not only hanging in there for the last super exciting talk. It is an absolute honor and a pleasure for me to have the chance to sit up here with Dr. Craig Venter. As you heard, Craig is actually not only an amazing visionary scientist, he's also an old boss of mine after he bought Cipher Genomics and I joined Human Longevity.
The key word is old.
No, previous boss of mine. But it really is an exciting day because for those of you who may not know much about the personal side of Craig, you actually have really deep roots here in San Diego. And so you've done just this incredible transformative science from right here. So maybe you could talk a little bit about that.
Well, I've been coming to San Diego. My grandparents lived here, so I started coming here as a kid in the summers. Growing up in the San Francisco Bay Area, the water for surfing was 53 degrees up there. So it was so much better to come down to San Diego. In fact, I graduated from high school in the Bay Area and left there when I was 17 and got drafted. I moved to Southern California to take up a surfing career and got literally drafted off my surfboard, but sent to boot camp in San Diego and then worked at Balboa Hospital for a long time running the infectious disease ward, which actually introduced me to most of the infectious agents that we ended up sequencing, and then followed it up with a year in Vietnam doing the same thing. So I got my bachelor's and PhD at UC San Diego. They tell me I still have, thank you. I have the shortest record for a PhD for the UC system, at least in biology. 3 years, and I published 21 papers in 3 years in Science and Nature and PNAS. So then moved out of state for a while.
Well, and so based on that, I think it's no surprise then that productivity translated into, your efforts to really break open genomics by leading the private effort to sequence the first human genome. And then it's not just about human genomics, which I want to make sure to come back to as well, but you've also done a lot of work in the microbiome. And so for you guys, if you're not aware, this book is Craig's latest book. It just came out this week, The Voyage of Sorcerer II. And I literally just got this off Amazon and have only cracked the first couple of pages, but it's a really exciting read about the voyage you took to really understand and unlock the biology of the microbiome of the ocean. And we heard from Rob Knight how powerful the microbiome is. So maybe you can tell us about that.
Well, it was based on our sequencing the first genome in 1995 and then doing some clinical isolates. We had a clinical isolate from Norway from a patient that was refractory from treatment. And on sequencing the agent, or the supposed agent, the algorithm was so powerful that it actually assembled 2 separate microorganisms. So at that stage, it was clear the algorithm that we developed was going to be so powerful and that each species had its own mathematical solution for its genome. After assembling 25 million fragments for the human genome. We knew that it was incredibly powerful, and we proposed to do a shotgun sequencing of ocean water, what was in ocean water, based on the assumption that prevailed in science at the time in the early 2000s, that there was very little diversity in the oceans. For example, as organisms were discovered, and discovery up until genomics was looking through a microscope or seeing what would grow in culture. If it didn't grow in culture, it didn't exist. And so in the Sargasso Sea off of Bermuda, they were up to the 11th microbe, SAR-11, for Sargasso Sea 11. And this just seemed totally absurd to me with the diversity of biology and the diversity of microbes. We now proved in this book that we are clearly a microbe planet. We discovered in the ocean more new species than there are stars in the universe. So it's been easy to recruit physicists into biology because they decide physics has, it's sort of a dumb field compared to biology, and biology has many more challenges. But that diversity is important. Half our oxygen we breathe comes from these organisms. And off the coast here, there's now multiple mile wide dead zones. The largest ones are the size of Africa, and they're getting larger. There's just an article in the New York Times in England to keep rivers alive. They're having to pump oxygen into the rivers because of all the pollution and all the dead zones. So everybody's worried about global warming and sea level rise. The biggest problem is going to be we could suffocate ourselves by slowly killing off our main oxygen producers. But it's a story about sailing 65,000 miles around the ocean and all the problems with dealing with politics of every country. We were arrested 3 times, 2 times held at gunpoint, once by the French and once by the British. The French Navy threatened to sink our sailboat. This was not too long after they sunk the Rainbow Warrior, the Greenpeace vessel in New Zealand from protesting their nuclear testing. They were worried we were going to sail to the Komodo Islands where they were doing nuclear testing and prove genetically how much they were damaging the environment. The U.S. Navy had to ultimately intervene and say that we were a U.S. protected vessel, and they just decided not to sink us. But if you like sailing stories combined with science, you'll enjoy the book.
I love it. I'm excited to read it. I think we're so fortunate to have incredible waters off the coast here, right in San Diego, and understanding what makes them thrive as a scientist is also really interesting.
Well, the people that are surfers or swimmers know that no locals go into the water here after it rains. So all the people out in the water are dumb visitors that a certain portion of them get sick from the outflow of all the sewage from the Tijuana River, plus these dead zones, plus the pollution is causing an increase in some toxic diatoms that literally cause birds and seals and humans, if they ingest this, to go a little bit insane. So it is changing, and hopefully this book will help make people more aware of it.
No, I love that. And for those of you I haven't met yet, I am at Illumina, so a company that develops a lot of the sequencing technology that enables DNA sequencing, and our primary mission is really using genomics to improve human health and planetary health, right? And so we've touched a little bit on the role that, understanding the sequence of all the organisms in the ocean can play to inform diversity and conservation efforts. Let's turn to the human health side of things, right? Because even if you're not in the field, I'm sure many of you remember that exciting announcement on the front lawn of, the White House when the first human genome sequences were published, and you were there with Francis Collins and Bill Clinton, and it was really heralding in this new era of precision medicine. So we're 22 years later, right? And I think we've made a lot of progress, but it's also really exciting to think about what's next. So you've had a couple of companies in this space, and we talked about human longevity, which has a very ambitious, vision of utilizing the genome to inform care. So maybe you can tell us about that.
So because sequencing became so dominant and because it changed so fast, it's now, our first genome cost us $100 million to do. You can now get your genome sequenced for roughly $200, but it's a competitor of Illumina that does it. So
we do it for $200 on the NovaSeq X. Very excited.
So there's been a huge focus on sequencing, and it's easy to do and enumerate, but just sequencing genomes has not been all that useful other than tracing ancestry and lineages. And so I felt for a long time that if we weren't doing extensive phenotype testing along with the genome, we were not going to make progress. And it's very hard to convince people of that. From my genome, there's been hundreds of papers published on all the different diseases I'm going to die from or should have died from. But one prediction that everybody keeps going back to is I'm heterozygote for ApoE4, and supposedly I should have Alzheimer's disease by now. I can't remember if I do. But I asked my friend who was head of neurology if I could get a brain scan and a PET MRI. And they refused to do it, saying it would be wrong for them to do it because I wasn't exhibiting any symptoms. But I said, I have this genetic information, we need to know if it means anything. So they finally agreed to do it under a pseudonym. And they said, pick a name. I said, how about Charles Darwin? And they said, no, that's too obvious. So they made up some phony name. But they called me a few weeks later and said that I had a brain of a 40-year-old. There was no trace of amyloid. My wife disagreed, said he has a brain of a 17-year-old. But I was relieved to know that these predictions, at least in my case, didn't mean anything. Now we've done hundreds and hundreds of individuals that are heterozygote for ApoE, and almost never do they show amyloid or Alzheimer's symptoms. There's more of a correlation with the homozygotes, but only with severe disease. And so it's very much like the so-called breast cancer genes. They at best give you a 50-50 lifetime chance of having breast or ovarian cancer. But that's not what people are usually told. The only thing that changes that is if you have a family history, every woman in your family has breast or ovarian cancer, then that plus the BRCA1 or BRCA2 takes it up into the high 90%. Which means BRCA1 and BRCA2 are not the causes of breast or ovarian cancer. They're linked to it loosely, and we still don't know the actual causes and these the absolute genetic conditions. So the same as with ApoE and a lot of other diseases. When these, quote, disease genes were discovered, they looked only at populations with the disease. And as soon as you go out to the broader population of the world, that doesn't work. And we learned that early on. We had actually, from 2 hospital studies we were doing in England, we had a fantastic correlation with a genetic marker in drugs used to treat breast cancer. And they appeared to be incredibly successful at predicting these treatments. And we had the paper already basically accepted in Nature. And I thought, maybe we should do a little broader testing. As soon as we went out to the broader, uninvolved environment, it turns out it was a fairly common allele that predicted nothing. And that's what happens, Darwin knew this, when you have isolated genetic lineages, you get a unique answer. But that doesn't mean they translate to the whole population. So that's led to the start of HLI, of doing comprehensive MRI in conjunction with the human genome. And we took it a whole lot further, and it got better when Ashley's team joined us. But basically, from all the phenotype testing and whole body MRI with new algorithms, we only looked at people that were, quote, healthy. But 5% of everybody over 50, we found a major tumor that they were unaware that they had. 1% of the entire population has a brain aneurysm that they were unaware of. But the data was skewed to a younger population, which means the older population didn't have it because they had already died off from their brain aneurysms. So a lot of these, they can be treated now as an outpatient, but a lot of these were so large, they were in danger of bursting, even in young people in their 20s. So we're finding a lot of things because of the new tools and the new algorithms in MRI, the new metabolomics, mixing it with all these other testings, and with having a much richer genome population. And what we're trying to do at the Venture Institute now is build a new version of HLI based on mobile labs to go out to indigenous populations and underserved communities where they wouldn't have to pay for the testing, but helping them get into where they would get treatment for discovered things. But getting back to the isolated populations, these populations, many of them have unique types of liver disease and others that we think by characterizing these populations with sequence and phenotyping, it's going to lead to new therapeutic discovery at the same time. So that's where we're trying to go with this next phase.
Well, and I think that's one of the things I found so remarkable about the human longevity concept is, if you're in the field, and I know many of you are from tech or other, parts of the ecosystem, if you're in genomics and life science, you're Mr. DNA, you're Mr. Genome. And so to recognize that you actually want to put all of this other phenotypic and kind of clinical data and imaging data together with the genome and add all these tools to me was pretty insightful and remarkable that we have more discovery left to do. And how do you?
I would say about 99% of discovery remains to be made because there's so little phenotype testing along with the genome. The late Tom Caskey was the world's best human geneticist, and we developed new AI tools that would search allele frequency in patients where we had this phenotype data, and he would make almost a new discovery link on a daily basis between the phenotype data and the patient's disease. This actually happened in my own case. I was testing out the first MRI machines after being told by UCSD radiology that I did not have prostate cancer. After I finished the test, our head radiologist came and told me that I had high-grade prostate cancer. It got re-biopsied. The biopsies, which are random, just totally missed. I had 2 very rapid growing tumors that I had removed 3 weeks later. Had I waited 6 months, they would have metastasized. So random checking and luck helps. But in fact, we couldn't figure out why I had such fast growing tumors after being told that, in fact, I was cancer-free. It turns out the testosterone receptor is a triplet repeat gene. So the fewer triplet repeats you have, the more receptor expression. People with a large number of triplet repeats are basically sterile. So you can give them all the testosterone in the world and it does nothing because there's no receptor. Well, it turns out I had the shortest number of triplet repeats known. I've always been accused of having way too much testosterone my whole life. Maybe
why you almost got sunk and shot and arrested, but who knows?
I was bald at age 21 and that was just one of the physical symptoms of too much. But in fact, on clinical testing, I had very low testosterone levels. And so the brilliant endocrinologist gave me exogenous testosterone. but because I had maximum number of receptors, I only needed a tiny bit of testosterone to maximally activate it. So being given more testosterone was like just pouring gasoline on a fire. We actually cultured the cells from my tumors. They grow 10 times faster in the presence of testosterone than without it. But it was a case of asymptomatic testing, and we discover at least one person a day with a major tumor. There's probably been 200 men in the San Diego area that we've discovered a major version of prostate cancer that they were unaware that they had, but many other kinds of tumors. And about 50% of people going through, we find some major disease. And so this is where we hope medicine will ultimately go, with a combination of pre-symptomatic testing. All the tumors we find are basically at stage 0, stage 1. And to my knowledge, they've all been completely treated. When you find them, because you have symptoms, they're at stage 3 at least, and your chance of survival goes down exponentially. So combined with new DNA tests that Illumina and others are doing, this field has a chance of changing.
I think that that's really the most exciting part about where we are in the history is, it's taken a long time to get to this point, but now with the reduction in cost of the tools, increase in accessibility, moving into underserved communities that aren't part of the research cohort, I think that we're going to accelerate that transition to precision medicine, right? Even faster. So I could sit here and talk to you all day. I've got a million questions. I want to make sure we get questions from everybody else as well. So yes, I think we have a question here.
Sure. Fred, you started your extraordinary academic and entrepreneurial career at a community college, College of San Mateo. How did that influence your future path? Well, it was critical, and I highly recommend it. So when I got out of Vietnam, because I barely graduated from high school because I had other interests, and I have aphantasia, so it's the opposite of having a photographic memory. I had to start school over from scratch. I was actually introduced probably to a better education at a community college for the first 2 years than you get at most universities. And the nice thing about the California community college is if you have a B average or better in transfer courses, the University of California has to accept you. So that's how I got into UC San Diego. I just had fantastic teachers that took, I would say, a special interest in my case, being a Vietnam veteran. So I had a better education at a community college than I probably could have gotten at any Ivy League school or any other school. So and it stood me well for rapidly going through UC San Diego. So thank you for asking.
Any other questions? One more question? Yes.
So with the 50% chances of some sort of a major disease that you mentioned, that people you have screens that had that high number of major diseases, how many of those were you actually?
So the question was, of the 50% of a major disease that we found in the, quote, healthy population, how many of those were treatable? Most of them, but not all of them, because we discovered, some risk of early onset diseases that they were already developing some indications for, that there were no good treatments. But there are some that were fantastic. And different people go through the health nucleus at HLI for different reasons. My mother-in-law was worried because her mother had just died from severe Alzheimer's, so she wanted to find out from the MRI scan whether she had Alzheimer's disease. She didn't. She has a much larger hippocampus than I do, and she never forgets anything. But we found out that she had an autoimmune liver disease that she was completely unaware of, but we found it at such an early stage by stopping to drink any alcohol and starting a treatment. The disease has not progressed. So we have success story after success story like that, including many people that have gone from clinic to clinic trying to find out what was wrong with them. But, unfortunately, all diseases are not currently treatable, including Alzheimer's disease, but most things are preventable with early diagnosis and early detection. So thank you for the question.
All right, and with that, we have a very exciting day ahead. So I want to thank Craig so much for joining and for your time and sharing your stories. You'll be around for a little bit longer if folks can find you.
If the bar is open.
Okay, so we'll get that
make
taken
sure.
care of. Thank you.
Thank you.
Thank you, guys. That was amazing.
I really feel passionate about sharing the San Diego talent, elevating San Diego, because it is the best place to be an entrepreneur.